Hi, everyone, and welcome to this edition of Budding Solutions, a series on disease and pest management in cannabis sativa. My name is Kim Leonberger, and I'm an Extension Associate in the Plant Pathology Department at the University of Kentucky. In this episode, we are going to take a closer look at beet curly-top virus. To discuss this important disease issue, I would like to introduce Dr. Punya Nacchapa. Punya received her Master's of Science degree in entomology from University of Georgia and a PhD in entomology from Kansas State University. After completing her PhD, she was a postdoctoral researcher at K-State and Texas A&M. Currently, she is an associate professor at Colorado State University. Her research program focuses on understanding the interactions between plants and insect vectors to manage plant pests and diseases in hemp and other cropping systems. She has a dynamic lab group with 2 postdocs, 2 research associates, 3 PhD students, 3 master's students, and several undergraduate students. Her research is funded by USDA NIFA and FFAR, as well as other commodity groups. Punya is going to share a few slides with us about this disease, then we'll have time for a few questions. Welcome, Punya.
Thank you once again, Kim. Today I'll be talking about the ecology, epidemiology, and management of curly-top disease transmitted by beet curly-top virus in hemp. So my lab website information is down at the slide, and at the end I have some resources about BCTV as well. So beet curly-top virus belongs to a group of small DNA viruses in the family Gemini-viridae in the genus Curtovirus. This virus causes curly-top disease, which has impacted agriculture in the Western United States since the early 1900s. The main crop that it impacts here are sugar beets, as you can see on the right panel, the infected plants compared to the healthy plants. The virus has a large or an extensive host range, including over 300 species in 44 plant families. Some important crops that it affects besides sugar beets are solanaceous crops like tomatoes. You can see the infected plant there. Another host species I should mention are weeds, which is going to be very important for the transmission of the epidemiology, which I'll talk about in the coming slides. In 2020, the disease was first reported here in Colorado in Delta County to be infecting hemp. And you can see a picture of an infected hemp plant here compared to a healthy plant. So my lab group and another group from Colorado had reported this. The symptoms are pretty spectacular and a wide range of symptoms. We presume this is to do with the different cultivars or genotypes that we see them on and environmental conditions. So you can see this flat stem phenotype here. Maybe I could put my pointer on, then you can see, this really distinct flat stem. This is more a typical curly top disease symptoms with the yellowing and the stunting. You can see some curling and mottling on this plant here. Here, the edges of the leaf sort of curved inward. My collaborator at that time, Dr. Whitney Cranshaw, was calling this a taco leaf phenotype. This is interesting where the middle or the central part of the branches are infected with the yellowing and the curling, but seems like the lateral branches had outgrown the symptom and looked healthy, and more curling and twisting here. Since its initial detection in Colorado, BCTV has been detected in just about every hemp growing state in the Western United States. I know there has been some anecdotal evidence in the Midwest. One of our collaborators was also reporting it in Wisconsin, but nothing's been confirmed as yet. We did a study to look at the impact of BCTV on cannabinoids. We did it in a high cannabinoid line and a low cannabinoid line, and you can see the different major cannabinoids here. And between the healthy and the infected plant, there was no impact on the cannabinoids, which is good news for our CBD growers. But we did find significant impact on biomass and flower yield. You can see a picture here. This is one of our grower collaborators, John House, with the BCTV infected plant compared to a healthy plant. Other states like Arizona has reported 100% loss in the crop. And just this, I should say last year, one of our field sites, we had over 50% infection and loss of the crop. So the impact can vary depending upon the variety and the location. Coming back to the virus a little bit, the virus is a phloem-limited virus, and it's characterized by a monopartite or a single nucleic acid genome. In this case, it is single-stranded DNA molecule encapsulated between two twin icosahedral or spherical particles, capsids. This is very characteristics of Gemini viridae. There are 11 different BCTV strains based on the nucleotide identity and host specificity. In hemp, the most common one we've detected are BCTV Worland and BCTV Colorado, but there are new strains emerging such as pepper curly top and pepper yellow dwarf. This phylogenetic tree that you see here are sequences of BCTV from hemp. The ones indicated in red are what we have in our samples, and these include all the Western states. And this is the whole genome of BCTV. And you can see they are clustering with BCTV Worland, BCTV Colorado, BCTV Curly Top, and BCTV Yellow Dwarf here. The ones indicated in the asterisks are the sequences available, whole genome sequences available in GenBank. The virus is exclusively transmitted by the beet leafhopper Circulifer tenellus. You can see the picture here in North America. But in Europe, in the Middle East, and the Mediterranean region, there's another species of Circulifer that can transmit BCTV. The way the virus is transmitted is that the insect, which also is a phloem-feeding insect, so the insect picks up the virus particles from the phloem. The virus circulates through the insect gut and then is transmitted to a healthy plant through the saliva. The virus does not replicate in the insect, so it's called persistent because it moves through the gut, circulative, but it does not replicate. Now, the bad news is that we've collected several different species of leafhoppers in hemp, and beet leafhopper, Circulifer tenellus, is not the most common. It happens to be Ceratigalia, which looks very similar to the beet leafhopper. One way to identify or differentiate between the different species is by the shape of the head. Beet leaf hoppers have more of a root-like shape, whereas Ceratigalia, you can see here, is more rounded and oval. Beet leaf hoppers don't have any markings on their face or the vertex, and you can see some here on this leaf hopper. Also, if you have a microscope, the abdominal segments are quite distinctive. Males have a more squarish terminal abdominal segments, and females are more rounded. But again, this is quite subjective. So it's I think the best option for growers would be to send the specimens off to a diagnostic clinic or your university extension specialist to confirm that it is in fact beet leaf hopper. So the life cycle of the virus is closely tied with the leaf hopper because that's how the virus spreads. To date, we don't know any other modes of transmission. There was one report of seed transmission of BCTV in Petunia. We've been doing some experiments in our lab, but it's too early to confirm. So the cycle of the virus, of course, starts with the leafhopper. Mated females typically overwinter on winter annuals like dandelion and prickly lettuce and perennial weeds like London rocket. And then in spring, the females will lay eggs and the nymphs hatch, and the nymphs or the offsprings pick up the virus from these winter annuals. And as these weeds dry down, the new generation move on to summer annual hosts like weed species such as kochia and Russian thistle, which they really love and they breed really well on it. They move on to important crops like sugar beet. And this is when hemp gets infected as they are moving into the summer hosts. Multiple generations of the insects can be produced in the summer. The populations can build up pretty to pretty significant numbers. And then come fall, the females and the mated individuals move back into their winter host and the cycle continues. So in the US, there are these overwintering or spring breeding regions in the Western United States. For Colorado, it's believed that the leafhopper populations, the migrants, are coming in from New Mexico and Arizona. So the quality and the health of the population depends upon these winter annuals and perennial weeds in these other locations. We think there is a breeding population up in Utah and Colorado, but we've not actually confirmed that. Now, while hemp can get infected with the virus, it's not a reproductive host for the leafhoppers. This graph is showing the number of nymphs in viriliferous or virus-infected, BCTV infected leafhoppers and non-viriliferous or healthy leafhoppers. And you can see it's less than an average of 5, whereas on sugar beets, they can reproduce 3 times as many. So even though a host may not be a reproductive host for the leafhoppers can spread the virus, while it's passing through or just incidentally, and the plant can get infected. So in terms of BCTV management in hemp, you really need an integrated pest management or disease management approach. And this is true for any vector transmitted disease. It starts with monitoring for the insect vector. And for leafhoppers, beet leafhoppers in particular, yellow sticky cards has been the go-to. You can put sticky cards around your field, and depending upon the size of the field. It's also important to monitor for leafhoppers in the weeds because they actually love reproducing in the weeds compared to, as I said, a hemp. So sweeping can be done to know if they're in the weeds. And then for diagnostics for the virus, you have ELISA kits are available, PCRs. Agdia also has a portable isothermal, a rapid isothermal assay that you can actually purchase. It is on the more expensive side, but it is, on-site diagnostics, which is really helpful. But I would say reach out to your diagnostic clinic or companies that can do this for growers. Weed management is critical because weeds play such an important role in the disease cycle. And of course, for hemp, the options are limited. Last year, the first herbicide was approved for hemp. It's called ethafluralin. The brand name is Sonalam. So that's one option, but mechanical removal and hand weeding is another option. You could also plant hemp into, a mulch or a plastic cover, as you can see in the background here. This is a CBD hemp field here in Colorado. So weed management is critical. For the insect itself, there aren't any published studies on the efficacy of insecticides. And again, for hemp, it's essentially organic option. So if it's a bad year and you see a lot of leaf hoppers, then definitely I would recommend spraying organic insecticides. In other crops, cultural control has worked pretty well. My collaborator in New Mexico has tried kaolin clay. You can see that picture of that whitish substance that you can spray on the plants. This was done for peppers, and it significantly reduced curly top virus incidence. The kaolin clay prevents the leaf hoppers from landing on the plant. But the downside is that if there's a rain event, that can wash away the clay, and then new growth will not be able to, acquire the clay, the spray, so you'll have to redo it. But it hasn't been shown for hemp. But essentially, you need a whole spectrum of approaches to target this very complicated disease. So in terms of resources, I want to make a plug for our CSU Hemp Entomology website. We've just started this website, and it's more of a curation of all the resources available, including the one at University of Kentucky. For Beet curly top virus, my collaborators and I put together this really nice video. It's available in English and Spanish, and the link is here. I also would highlight the Oregon State University Extension website, especially for pests and diseases that is predominant in the Western United States. It's a great resource. But I think all of us are trying to acquire as many resources as possible and make it available for our growers. And with that, I want to thank, of course, my funding agencies, grower collaborators, and of course, my team, my students and postdocs that actually do all of this work that I get to present. And I will stop here and take any questions.
Excellent. Thank you so much, Punya. That was so informative. That's an area that I know so little about, so I really enjoyed learning. Couple of questions for you. So first, you talked a little bit about the biology of both the pathogen as well as the insect, and since we have both at play here, can you talk and explain a little bit more about how understanding the biology of the pathogen and the insect help in making management decisions?
Thank you, Kim. That is such an important question, and often, plant pathologists for a vectored, insect vectored virus forget about the insect aspect, and the entomologists forget that the insect is, within a few minutes to hours can transmit a pathogen. So one example is understanding, the population dynamics of the insect when it is moving into your area, and once it does, how does the population go? So this is critical, and when we have some studies. We're trying to do some studies here in Colorado. Once you know that you could then target insecticidal sprays if you know that the population is moving in let's say early to mid-May, target sprays for the insect. In terms of understanding the pathogen impact, I didn't highlight this too much, but in sugar beets we find that insects that are carrying the virus actually have a higher reproduction than insects that do not. And we see this mutualism in other virus vector relationships as well. So it's important for growers to know that you just can't let the population, go, because they will build up significantly virus infected individuals. But this also gives us an opportunity, and we've just published on this, is to look at how the virus is modifying the insect behavior and the physiology, and are there genes that we can then target to knock out or silence using RNAi sprays or genetic modification. Not right now for hemp, GMO is not allowed, but for something like sugar beets and tomatoes, that would be an option. So understand this interaction is really crucial from pest management and also, from the biology and behavior aspect of it.
That is really interesting. So I know that your lab has generated a huge body of work in this area, but what areas do you think are still lacking, or maybe areas that you'd want to focus on in the future as far as research?
Yes, my goodness. Yeah, my department head mentioned when I started working on this, and this virus has been around for several of hundreds of years, but not very well studied, and definitely not in hemp. And my department head mentioned that I have my career, set with just working on BCTV and BLH. One thing which is which is lacking, even for a crop like sugar beet, is no knowledge about resistant genes or resistant varieties. There is no good resistant varieties for sugar beet. There is a beet curly top resistant variety in beans, I believe, but not for sugar beets or for tomatoes and peppers. And so for hemp, I think given the limited, insecticide, herbicide options, knowing if there is screening hemp varieties and genotypes and finding something that has resistance, identifying the genes and being incorporating that into the commercial varieties, I think would be is important. I think genetic resistance is going to be important, and we've been working. I didn't have time to show that because of the time limitations, but we have been working with the hemp genetics company here. My students screened over 20. Actually, the paper will be coming out in Phytobiomes pretty soon. So she screened a panel of 20 hemp genotypes, and there were several, at least two or three genotypes that had significantly lower virus BCTV titer. So this is something that we could look into to understand the mechanisms and lead BCTV resistance, and maybe even try to move, understand insights in hemp could help us develop resistant varieties for sugar beets and other important crops as well. So I'm really excited about that.
That sounds really exciting. Last question for you. So this webinar series is all for growers. Would you mind to talk a little bit about how you connect growers to what's happening within your lab or at your institution related to hemp?
Oh, this has been probably the biggest challenge, especially for hemp. I work with other commodity groups, wheat, sugar beet, potato, that have been around, for much longer. Hemp is essentially a new crop here, and so we don't really have a commodity group. I'm leaning in my lab. We're leaning heavily on our Colorado Department of Agriculture to get some of our information out. We also have, as a lot of universities do, field days, not specifically for hemp, but and our agricultural experiment station workshops and conferences is where I get to present. We are just starting, as you mentioned before the presentation, we're starting to put out a newsletter to hemp growers and try to get some of this information out there and some of the resources I mentioned. But it has been a challenge, and I know the growers really are suffering because of the breakdown in lab to field transfer.
Absolutely. With a new crop, definitely a challenge trying to make sure that that information is getting into the right hands. But thank you so much for joining us today, Punya. I really appreciate it. This was very informative. Just a reminder to our viewers to please check the description below the video. We'll post all of the links that Punya identified there, as well as additional resources. And thank you again for joining us, and we hope to see you on the next episode of Budding Solutions.